Complex brain networks, graphtheoretical analysis of structural and functional systems by Ed Bullmore and Olaf Sporns. Abstract. Recent developments in the quantitative analysis of complex networks, based largely on graph theory, have been rapidly translated to studies of brain network organization. The brain's structural and functional systems have features of complex networks, such as small world topology, highly connected hubs, and modularity, both at the whole brain scale of human neuroimaging and at a cellular scale in non-human animals. In this article, we review studies investigating complex brain networks in diverse experimental modalities, including structural and functional MRI, diffusion tensor imaging, magnetoencephalography, and electroencephalography in humans, and provide an accessible introduction to the basic principles of graph theory. We also highlight some of the technical challenges and key questions to be addressed by future developments in this rapidly moving field. This paper, by the way, is I think this is where Olaf Sporns coined the term connectome. This is kind of the quintessential connectomics paper. So, graph theory, by the way, is a branch of mathematics that deals with the formal description and analysis of graphs. A graph, defined simply as a set of nodes, vertices, linked by connections, edges, and may be directed or undirected. When described a real-world system, a graph just provides an abstract representation of the system's elements and their interactions. The University of Cambridge Behavioral Clinical Neuroscience has developed a Department of Psychiatry at Edinburgh's Hospital, Cambridge, UK, GlaxoSmithKline. Clinical Unit, Cambridge, Department of Psychological and Brain Sciences, Indiana University, Bloomington, Indiana 47405, USA. Correspondence to email. So, we have known since the 19th century that neuronal elements of the brain constitute a formidably complicated structural network. Since the 20th century, it has also been widely appreciated that this anatomical substrate supports the dynamic emergence of coherent physiological activity, such as phase-locked, high-frequency electromagnetic oscillations that can span multiple spatially distinct brain regions that make up a functional network. Such networks are thought to provide the physiological basis for information processing and mental representation. In this article, we focus on graph theoretical approaches to the analysis of complex networks that could provide a powerful new way of quantifying the brain's structural and functional systems. Box 1. That's this. Structural functional brain networks. anatomical parcellation, the slices, the little slices go become these slices, and they're made into structural brain network, the recording sites, time series, and make the functional brain network, graph theoretical analysis. So we'll get to that. But since the mid 1990s, developments in our understanding of physics of complex systems have led to the rise of network science as a transdisciplinary effort to characterize network structure and function. In this body of literature, complexity arises in the macroscopic behavior of a system of interacting elements that combines the statistical randomness with regularity. The ascendancy of network science has been driven by the growing realization that the behavior of complex systems, as as be they society, cells, or brains, is shaped by interactions among the constituent elements. The increasing availability and tractability of large high quality data sets on a wide range of complex systems has led to the fundamental insight. Substantially different complex systems often share certain key organizational principles, and these can be quantitatively characterized by the same parameters. In other words, many complex systems show remarkably similar macroscopic behavior despite profound differences in microscopic details of the elements of each system or their mechanisms of interaction. Let's describe in box 2, by the way. In box 2 is this. Talks about the network measures, all those network measures, and some graphs. Get to that. One example of an apparently ubiquitous macroscopic behavior in complex systems is the small world phenomenon, box 3. Recently, small world architectures have been found in several empirical studies of structural and functional brain networks in humans and other animals. And over a wide range of scales in space and time. Small worldness is naturally, therefore, a key topic of this review. However, discovering that brain networks are small world networks is only the first step towards comprehensive understanding of how these networks are structurally organized and how they generate complex dynamics. In network science, methodological advances allow us to quantify other topological properties of complex systems, such as modularity, hierarchy, and centrality and distribution in network hubs, many of which have already been measured in brain networks. There have also been significant efforts to model the development or evolution of complex networks to link network topology to network dynamics and to explore network robustness. Yeah, okay. So we have here is a structural and functional brain networks can be explored using graph theory thoughts. You're using graph theory through the following four steps. See the figures. Define the network nodes. These could be defined as electroencephalography or multi-electrode array electrodes, or as anatomically defined regions of histological MRI or diffusion tensor images. That's anatomical parcellation or the recording, all both of those. Um, like the Neuralink product the Elon Musk's working on, or just really high quality MRIs. Um, Estimate a continuous measure of association between nodes. This could be the spatial coherence or Granger causality measures between two magnetoencephalography sensors, or the construction probability, the connection probability between two regions of an individual diffusion tensor imaging dataset, or the interregional correlations in cortical thickness or volume MRI measurements estimated in groups of subjects. Generate an association matrix by compiling all pairwise associations between nodes and usually apply a threshold to each element of this matrix to produce a binary adjacency matrix or unidirected graph. Let's see a picture of the adjacency matrix down here somewhere. Yeah, this is kind of an adjacency matrix. There, there. Calculate the network parameters of interest in this graphical model of a brain network and compare them to the equivalent parameters of a population of random networks. So, we compare these to random networks, then see how brain organization differs from random networks. Each step entails choices that can influence the final results and must be carefully informed by the experimental question. At step one, parcellation schemes can use prior anatomical criteria to be informed by the functional connectivity profiles of different regions. Several such parcellation schemes may be available and can affect network measures. In most magnetoencephalography and electroencephalography studies, network nodes are equivalent to individual electrodes or sensors, but networks could also be based on reconstruction anatomical sources. However, some reconstruction algorithms will determine the brain location of each of these sources by minimizing the covariance between sensors, which has major effects on the configuration of functional networks. At step two, a range of different coupling metrics can be estimated, including measures of both functional and effective connectivity. A crucial issue at step three is the choice of threshold used to generate an adjacency matrix from the association matrix. So a crucial issue, I'm going to highlight the threshold. Crucial threshold. So generate graphs of different sparsity or connection density. So the crucial issue of step 3 is a choice of threshold to generate an adjacency matrix from the association matrix. Different thresholds will generate graphs of different sparsity or connection density. And so network properties are often explored over a range of plausible thresholds. Finally, step 4, let's do step 3 and step 4, graphtheoretical analysis. There, a large number of network parameters can be quantified, box 2. These must be compared with the null distribution of equivalent parameters estimated in random networks containing the same number of nodes and connections. It's this random networks connected. Statistical testing of network parameters may best be conducted by permutation or resampling-based methods of nonparametric inference, given a lack of statistical theory concerning the distribution of most network metrics. Most graph theoretical network studies to date have used symmetrical measures of statistical association or functional connectivity, such as correlations, coherence, and mutual information, to construct unidirectional graphs. This undirected graphs. This approach could be generalized to consider asymmetrical measures of causal association or effective connectivity. Thus, uh, such as Granger causal or dynamic causal coefficients to construct directed graphs. It is also possible to avoid the thresholding step by analyzing weighted graphs that contain more information than the simpler unweighted and undirected graphs that have been the focus of attention to date. Structural brain network image is reproduced from Ref 59, functional brain images, reproduced in permission Ref 59, Society for Neuroscience. So the complex network is defined as an informal description of a network with certain topological features such as high clustering, small worldness, the presence of high degree nodes or hubs, assortativity, modularity, or hierarchy that are not typical of random graphs or regular lattices. Most real-life networks are complex by this definition, and analysis of complex networks therefore forms an important methodological tool for systems biology. And an adjacency matrix indicates the number of edges between each pair of nodes in a graph. For most brain networks, the adjacency matrix is specified in binary. That is, each element is either 1 if there is an edge between them, the nodes, or 0 if there is no edge. For undirected graphs, the adjacency matrix is symmetrical. Okay, so this was box 1. We left off reading the article. There's development, evolution, complex network to link network topology to dynamic networks and to explore network robustness and vulnerability. The topics to vulnerability is like if there's a hub, if it's taken out, then the network gets more disconnected. robustness and vulnerability. The topics are likely to become increasingly relevant in relation to neuroscience. In this article, we describe and discuss the expanding interface between systems, neuroscience, and the physics of complex networks. Review the existing empirical data on topological and dynamical properties of structural and functional brain networks and ask what this literature tells us about how structural networks shape functional brain dynamics. Space limitations prevent us from providing coverage of all animal models, in vitro uh, preparations, and human studies that have contributed to this endeavor. We necessarily focused on what we consider to be some representative examples of graph theoretical research in brain networks, with an emphasis on studies of the human brain. We thus consider the implications of complex brain networks for an understanding of neuropsychiatric disorders and conclude with some general remarks from the evolution of scale invariant topology in brain networks and some key features and challenges of this emerging field. Let's take a look at some more of these boxes. Let's take a look at box 2, network measures. A network is defined in graph theory as a set of nodes or vertices and the edges or lines between them. Graph topology can be equivalently quantitatively described by a wide variety of measures, some of which are discussed here. It is not yet established which measures are most appropriate for the analysis of brain networks. The figure shows a schematic diagram of a brain network drawn as a directed left and undirected right graph. Both structural and functional networks can be either directed or undirected, as shown in box 1. All right, so there's directed means there's arrows, undirected means there's no arrows. Node degree distribution, and assortativity. The degree of a node is the number of connections that link it to the rest of the network. This is the most fundamental network measure, and most of the measures are ultimately linked to node degree. The degrees of all the network's nodes form a degree distribution. In random networks, all connections are equally probable, resulting in a Gaussian and symmetrically centered degree distribution. Complex networks generally have a Gaussian degree distribution, often with a long tail towards high degrees. Highlighted. Complex networks generally have a non-Gaussian degree distribution, often with a long tail towards high degrees. Random networks are probably Gaussian distribution, symmetrically centered. The degree distributions of scale-free networks follow the power law. Assortativity is the correlation between the degrees of connected nodes. Positive assortativity indicates that high-degree nodes tend to connect to each other. That's kind of a rich thing. Clustering coefficient and motifs. If the nearest neighbors of a node are also directly connected to each other, they form a cluster. The clustering coefficient quantifies the number of connections that exist between the nearest neighbors of a node as a proportion of the maximum number of possible connections. Random networks have low average clustering, whereas complex networks have high clustering associated with high local efficiency of information transfer and robustness. Interactions between neighboring nodes can also be quantified by counting the occurrence of small motifs of interconnected nodes. The distribution of different motif classes in a network provides information about the types of local interactions that the network can support. Path length and efficiency. Path length is the minimum number of edges that must be traversed to go from one node to another. Random and complex networks have short mean path lengths or high global efficiency in parallel information transfer, whereas regular lattices have long mean path lengths. Efficiency is inversely related to path length, but is numerically easier to use to estimate the topological distances between elements of disconnected graphs. Connection density or cost. Connection density is the actual number of edges in the graph as a proportion of the total number of possible edges, and is the simplest estimator of the physical cost. For example, the energy or other resources required of a network. Now, hubs, centrality, and robustness. Hubs are nodes with the highest degree or high centrality. Centrality of a node measures how many of the shortest paths between all node pairs in a network pass through it. A node with the highest centrality is thus crucial to efficient communication. The importance of an individual node to network efficiency can be assessed by deleting it and examining and estimating the efficiency of the lesion network. Robustness refers to the structural integrity of the network following deletion of nodes or edges and perturbations on a local or global network states. Modularity. Many complex networks consist of a number of modules. There are various algorithms that estimate the modularity of a network, many of them based on hierarchical clustering. Each module contains several densely interconnected nodes, and there are relatively few connections between nodes in different modules. Hubs can therefore be described in terms of their role in this community structure. Provincial hubs are connected mainly to nodes of their own modules, whereas connector hubs are connected to nodes in other modules. There you go. Connector hub, it connects these two modules. A provincial hub connects all many nodes in its own module. Okay, so that was box 2. Box 3. Random and scale-free and small world networks. In random graphs, each pair of nodes has an equal probability, P, of being connected. Large random graphs have Gaussian degree distributions, as discussed in box 2. It is known that most graphs describing real-world networks significantly deviate from the simple random graph model. Some networks, including the Internet and the World Wide Web, have degree distributions in the form of a power law. That is, the probability that a node has degree k is given as probability k is proportional to k to the power of negative lambda. It's a power law. It's k to the power of something. In biological terms, the degree exponent lambda often ranges between 2 and 3. And is very gradual power law decay, and the degree distribution implies that the network lacks a characteristic scale. Hence, such networks are called scale-free networks. Barabelsi and Elbert demonstrated that scale-free networks can originate from a process by which each node that is added to a network as it grows connects preferentially to other nodes that already have high degree. Scale-free networks are unlikely to be if the attachment of connections is subject to physical constraints or associated with cost. Therefore, physically embedded node networks in which nodes have limited capacity for making connections often do not have pure power law distributions, but may instead demonstrate exponentially truncated power law degree distributions, which are associated with a lower probability of very high degree nodes. Originally described in social networks, the small world property combines high levels of local clustering among nodes as a network to form families or cliques, and short paths that globally link all nodes of the network. This means that all nodes of a large system are linked through relatively few intermediate steps, despite the fact that most nodes maintain only a few direct connections. Mostly within a clique of neighbors. Small world organization is intermediate between that of random networks, the short overall path lengths of which is associated with a low level of local clustering and that of regular networks for lattices. The high level of clustering in which is accompanied by a long path length. A convenient single number summary of small worldness is thus the ratio of the clustering coefficient to the path length. After both metrics have been standardized by comparing their values to those of equivalent random networks, evidence for small world attributes has been recorded in a wide range of studies of genetic, signaling communications, computational, and neural networks. These studies indicate that virtually all networks found in natural and technological systems have non-random, non-regular, or small world architectures, and that the ways in which these networks deviate from randomness reflect their specific functionality. All right, so let's talk about connectivity real quick. In the brain, connectivity can be described as structural, functional, or effective. Structural connectivity denotes a network of anatomical links. Functional connectivity denotes a symmetrical statistical association or dependency between elements of the system, and effective connectivity denotes directed or causal relationships between elements. A microcircuit, a neuronal network composed of specific cell types and synaptic connections, often arranged in a modular architecture and capable of generating functional outputs. And the connectome, the complete description of the structural connections between elements of a nervous system. So the structural brain networks, topological and physical properties of structural networks. The anatomical configuration of brain networks ranging from interneuronal connectivity to interregional connectivity has long been a focus of empirical neuroscience. Network analysis, and in particular graph theory anatomical graph theory in box 4, there's box 4, origins of graph theory, offers new ways to quantitatively characterize anatomical patterns. According to graph theory, structural brain networks can be described as graphs that are composed of nodes, vertices, denoted neural elements, neurons, or brain regions that are linked by edges representing physical connections, synapses, or axonal projections. Although graph theory emphasizes topological connectivity patterns, the topological and physical distances between elements in brain networks are often intricately related. The neurons and brain regions that are spatially close have a relatively high probability of being connected, whereas connections between spatially remote neurons or brain regions are less likely. Longer axonal projections are more expensive in terms of their material and energy costs. It has been suggested that the spatial layout of neurons or brain regions is economically arranged to minimize axonal volume. Thus, conservation of wiring costs is likely to be an important selection pressure on the evolution of brain networks. Mapping structural networks in animal models. Tracing individual neuronal processes has been a long-standing technological challenge, and few structural brain networks have been mapped at cellular resolution. Serial reconstruction, or of electron microscopy sections allowed the complete connection matrix of the nematode C. elegans to be visualized. Currently, there is only one nervous system to have been comprehensively mapped at a cellular level, and it was the first to be described as a small world network. In larger brains, combinations of physiological and anatomical techniques uh, have allowed patterns of neuronal connections to be identified, leading to the identification of neuronal microcircuits and the formulation of probabilistic connection rules. Small world properties have been demonstrated in biologically accurate models of cellular networks in the reticular formation of the vertebrate brainstem. Reconstruction of cellular networks in mammalian neocortex from multi-electrode activity recordings has revealed several highly non-random features of connectivity, including a tendency for synaptic connections to be reciprocal and clustered. A promising approach for mapping connectivity involves the stochastic expression of several fluorescent proteins. This may ultimately deliver a complete map of the cellular interconnections of an entire brain. Simple fluorescent proteins may ultimately deliver a complete. Histological dissection and staining degeneration methods and axonal tracing have been used to map cerebral white matter connections. The pathways identified by these methodologies have formed the basis for systematic collection of species-specific anatomical connection matrices, including those for the macaque visual cortex, cat thalamocortical system. Network analysis of such data sets demonstrate high clustering of functionally related areas with short average path lengths, hallmarks of a small world architecture. Clusters identified by network analysis map on to known functional subdivisions of the cortex. Long-standing cortical projections facilitate short path communication despite increasing axonal volume. The cortical networks of several mammalian species also consistently demonstrated an overabundance of motif classes associated with network modularity and functionally diverse circuitry. Let's take a look at box 4, the origins of graph theory. Graph theory is rooted in the physical world. In 1736, Euler showed that it was impossible to traverse the city of Königsberg's 7 bridges across River Pragal exactly once and return to the starting point. To prove the conjecture, Euler represented the problem as a graph, and this original publication is generally taken to be the origin of a new branch of mathematics called graph theory. In the middle of the 19th century, the analysis of electrical circuits and the exploration of chemical isomers led to the discovery of additional graph theoretic concepts. Today, graph theory pervades many areas of science. Significant progress in graph theory has come from the study of social networks. One prominent experiment tracked paths of acquaintanceship across a large social network and found that that even very large networks could be traversed on average in a small number of steps. Although the small world phenomenon quickly captured the public imagination, its origins remained obscure until its association with specific types of connectivity was demonstrated. Box 3. The dual discoveries of small world and scale-free networks launched the modern era of graph theory, which now extends into biology and neuroscience. Neural systems have long been described as discrete elements linked by connections. Nevertheless, graph theory has essentially only been applied to neuroscience in the last 10 years. All right, mapping structural networks in the human brain. Several attempts have been made to map the structural networks of the human brain, also known as human connectome. One study derived structural connectome patterns from cross correlations in cortical thickness or volume across individual brains, which might directly indicate the presence of cortical pathways. Graph analysis revealed small world attributes and the existence of local communities of brain regions. Now, that's important. I'll highlight that. A more detailed analysis of the modularity or community structure of connection data sets derived from cortical thickness correlations revealed significant overlap between anatomical network modules and functional areas in the cortex. Human brain structural networks have also been mapped using diffusion imaging and tractography. A map of 70 to 90 cortical and basal brain gray matter areas was constructed using diffusion tensor imaging, or DTI, and analyzed using methods from graph theory. The network exhibited high clustering and short path length, and contained motif classes similar to those identified from tractography data. Several areas, including the precuneus, the insula, and superior parietal cortex, and the superior frontal cortex, were found to have high betweenness centrality and thus to constitute putative hubs. Another study that mapped connections between 78 cortical regions using DTI also identified several hub regions, including the precuneus and the superior frontal gyrus. Due to the limitations in the model that is used to infer fiber bundle orientation, DTI has difficulty detecting crossing fiber bundles. Diffusion spectrum imaging can overcome this limitation by constructing multiple diffusion directions in each voxel, and was used to build cortical connection matrices between 500 and 4,000 homogeneously distributed regions of interest. Again, network analysis revealed small world architecture of the cortical network. A more extensive analysis of 998 regions of interest networks obtained from 5 participants identified structural models interconnected by highly central hub regions. When considering multiple network measures, including node degree, connection, strength, and centrality, a particular set of brain regions located predominantly in the posterior medial cortex, including portions of the posterior cingulate and precuneus, was highly and densely interconnected, forming a structural core. Although they differ in terms of their experimental methodology and cortical parcellation, most of these studies revealed highly clustered, large-scale cortical networks. And most pathways existing between areas that are spatially close and functionally related. These clusters or modules are interlinked by specialized hub regions, ensuring the overall path lengths across the network are short. Most studies identified hubs among parietal and prefrontal regions, providing a potential explanation for their well-documented activation by many cognitive functions. Particularly notable is the prominent structural role of the precuneus, a region that is homologous to the highly connected posteromedial cortex in the macaque. The precuneus is involved in self-referential processing, imagery, and memory, and its deactivation is associated with anesthetic-induced loss of consciousness. An intriguing hypothesis suggests that these functional aspects can be explained on the basis of its high centrality in the cortical network. And diffusion tensor imaging is the DTI is an MRI technique that takes advantage of the restricted diffusion of water through myelinated nerve fibers in the brain to map the anatomical connectivity between areas. Then diffusion spectrum imaging is an MRI technique that's similar to DTI, but with the added capability of resolving multiple directions of diffusion in each voxel of white matter. This allows multiple groups of fibers at each location, including intersecting fiber pathways, to be mapped. And then cortical parcellation is a division of the continuous cortical sheath into discrete areas or regions. Brodmann's division of the cortex into areas defined by their cytoarchitectonic criterion is the most famous, but not the only parcellation scheme. And neuronographic measurements are recordings of epileptiform electric activity at specific sites of the cortex following topical application of a proconvulsive drug to a distant cortical site. Rapid propagation of electrical activity from stimulation to recording sites implies that the sites are anatomically connected. And then a functional MRI is the detection of changes in the brain region activity through their effects on blood flow and blood oxygenation, which in turn affects magnetic susceptibility and tissue contrast in magnetic resonance images. Then electroencephalography, or EEG, is a technique used to measure neural activity by monitoring electrical signals from the brain, usually through scalp electrodes. EEG has good temporal resolution, but relatively poor spatial resolution. Then magnetoencephalography, MEG, is a method of measuring brain activity by detecting minute perturbations in the extracranial magnetic field that are generated by the electrical activity of neuronal populations. Functional brain networks. Although analyzing structural networks helps us to understand the fundamental architecture of interregional connections, we must also consider functional networks directly to elucidate how this architecture supports neurophysiological dynamics. Despite considerable heterogeneity in the methodological approaches, there is an encouraging degree of convergence between studies of functional brain networks. The first such study used a set of neuroimaging measurements of the propagation of epileptiform activity following localized applications of strychnine to the macaque cortex. This, yeah, the macaque cortex. It's This demonstrated the pattern of functional connections between cortical areas that was consistent with a small world network. As we discussed below, these findings have been extended by studies based on functional MRI and electroencephalography, e.g., magnetoencephalography, MEG, or multi-electrode array, MEA data. Although such studies based on graph theory are the focus of this review, we note that other methods to investigate brain functional systems have recently been developed, including mathematical models of effective connectivity between regions. Effective connectivity models such as structural equation modeling, dynamic causal modeling, or Granger causality involve estimating the causal influence that each element of a system exerts on the behavior of other elements. Uh, Thus measures the effective connectivity between multiple regions can be used to generate a directed graph, which can then be topologically described using graph theory. However, the functional network studies reviewed below have all been based on undirected graphs derived from simpler measures of functional connectivity or symmetrical statistical association between brain regions. The key point is that, in principle, graph theory could be applied to an association matrix of either functional or effective connectivity measures to generate either undirected or directed graphs, respectively, although all neuroscientific studies to date have in fact been based on the measurements of functional connectivity. Now, mapping functional networks using fMRI. fMRI, functional MRI. Yeah, yes, that's for functional networks. Yeah, yes, and use that for mapping functional networks in the brain rather than structural ones. So the first graph theoretical study of fMRI data measured the partial correlations of resting state blood oxygen dependent bold signals between 90 cortical and subcortical regions that reported small world properties of the resulting whole brain network. Almost simultaneously, another study reported small world properties of functional networks derived from a set of activated voxels in fMRI data. The voxel level network was also reported to have a scale-free degree distribution. Subsequently, small world properties with parameter values similar to those previously reported in topological studies of cat and macaque and anatomical connectivity matrices were confirmed in a low frequency 0.03 to 0.06 hertz whole brain network derived from a wavelet correlations between regional mean time series. The high degree nodes or hubs of this network were mostly regions of multimodal association cortex in the degree distribution of an exponentially truncated power law. Other studies have explored the community structure of fMRI networks uh, using a hierarchical clustering analysis and showed that functionally and or anatomically related brain regions are more densely interconnected with relatively fewer connections between functional areas. Again, echoing prior work on anatomical connectivity matrices, the high density of connections between functionally related regions increases the clustering coefficient of the graph, whereas the long-range connections between different modules or clusters, even though they are relatively few in number, keep the path length low. Thus, the small world architecture of a brain functional network is closely related to the modularity. There are several other metrics for quantifying small world architecture in brain functional networks. Studies in statistical physics have shown that path length is inversely related to the global efficiency of a network for transfer of information between nodes by multiple parallel paths. And that global efficiency is easier to estimate than path length when studying sparse networks. Furthermore, the clustering coefficient can be regarded as a measure of the local efficiency of information transfer or the robustness of a network to deletion of individual nodes. The structural network of the macaque brain was found to have high global and local efficiency and to be sparsely connected. Thus, the macaque brain has economical small world properties. It has high global efficiency of parallel information transfer and high local fault tolerance for relatively low connection density. These concepts were translated to the analysis of resting state fMRI data acquired from young and elderly adults using the wavelet correlation, a measure of association between time series and of a specific frequency band to estimate the functional connectivity between regional bold time series endogenously oscillating in the frequency interval 0.06 to zero point 0.1 hertz For a more detailed review of the rationale for wavelet analysis in fMRI, see reference 78. In younger adults, functional brain networks demonstrated small world properties over a broad range of connection densities or costs. Relatively sparse networks were associated with maximum cost efficiency. The older adult group also showed evidence of small world properties, but had significantly reduced cost efficiency. They had to be relatively overconnected to provide the efficiency of parallel information transfer seen in younger brain network. The suggestion that aging is associated with changes in the economical small world properties of brain functional networks converges with studies that show differences in attentional and default mode networks between children and young adults. Normal processes of brain maturation and senescence might thus be reflected in quantifiable changes in functional network topology. Um, um, so yeah, we have a multi-electrode array is a technique for simultaneously measuring the electrical activity of local populations or single neurons, usually in tissue slices or cell cultures in vitro. Um, then an association matrix is a matrix that represents the strength of associations between each pair of nodes in a graph. Association between nodes can be quantified by many continuously variable metrics such as correlation or mutual information. Either functional or effective connectivity measures can be used to construct an association matrix. The blood oxygen level dependent bold signals changes in the magnetic susceptibility and MRI tissue contrast that are indirectly indicative of underlying changes in spontaneous or experimentally controlled brain activation. And then the default mode network is a set of brain regions including medial, frontal, and posterior cingulate areas of the cortex that are consistently deactivated during the performance of diverse cognitive tasks. Now, the mapping functional networks using electrophysiological techniques. When comparing the results of fMRI studies to results obtained using electrophysiological techniques like EEG, MEG, or MEA, many aspects of the data clearly differ. fMRI has good spatial resolution on the order of millimeters, but poor temporal resolution on the order of seconds, restricting the measurable bandwidth to approximately 0.001 to zero point 0.5 hertz. And fMRI measures activation-related hemodynamics rather than neuronal activation per se. All electrophysiological methods measure neuronal activity more directly and have better temporal resolution, with bandwidth typically 1 to 100 hertz, but they often have worse spatial resolution on the order of millimeters or centimeters for MEG and EEG, or less complete anatomical coverage in the case of MEA than fMRI. Another point of difference is that the nodes of the network derived from fMRI data will be anatomically localized regions or voxels of the image, whereas the nodes of a network derived from MEG or EEG data could be the surface sensors or recording electrodes. However, we can compare the topologies of networks derived from these different data sets by invoking a general operating principle of complex network analysis. Microscopically distinct systems can be informally compared in terms of their macroscopic organization using graph theory. Functional connectivity between pairs of electrodes has been estimated using the measures of generalized synchronization. And then thresholded to generate functional networks. In several studies of EEG and MEG data sets, this has shown that small world topology is represented in many frequency intervals and can be related to cognitive performance and normal aging. An alternative approach used wavelength correlations to estimate frequency-dependent functional connectivity between MEG sensors, again revealing that many topological and dynamic properties of brain functional networks were conserved across frequencies. This timescale invariance of wavelength correlations and the brain functional network parameters derived from them is a theoretically predictable corollary of the long-range autocorrelations of neurophysiological time series. Conservation of functional network properties. There have been fewer studies of functional networks in non-human species. In anesthetized rats, fMRI has been used to demonstrate small world and module properties of whole brain functional networks. A recent study considered functional coupling between pairs of cortical neurons measured by multi-unit electrodes in the visual cortex of an anesthetized cat. Most of the properties of these neuronal interaction networks could be accounted for by the pairwise correlations between electrodes, and the networks had small world properties with some highly connected hub neurons. The small world organization of functional networks at the cellular level has also been described on the basis of MEA recordings from in vitro cultures of cortical neurons. The MEA, that's a multi-electrode array. The results indicate that small worldness might be a conserved property of brain functional networks over different species and spatial scales. So preliminary evidence suggests that other topological properties of brain functional networks, such as the degree distribution, might also be conserved over spatial and temporal scales. Many large networks exhibited scale-free power law degree distribution, indicative of the existence of highly connected nodes. Box 3. Some studies of functional brain networks carried out at high spatial resolution, single voxels in fMRI, have provided evidence of scale-free organization. However, in other studies of functional networks derived from fMRI and MEG datasets over a wide range of frequency intervals, the empirical degree distribution conforms to exponentially truncated power law, implying the probability of highly connected node or hub is greater than an individual random network, but less than would be expected in a scale-free network. Truncated power law degree distributions have also been reported from analysis of anatomical networks derived from structural MRI data in humans and from analysis of functional networks derived from MEA data in cats, multi-left array. These distributions are typical of physically embedded networks, such as the global air transportation network, in which the maximum degree is limited by physical considerations such as finite capacity of any node to receive connections. The reason for the differences between these findings and reports of scale-free properties are currently unknown. However, it is notable that pure power law scaling of the degree distribution of human brain functional network has only been reported by voxel level analysis, whereas exponentially truncated power laws have been reported by regional level analysis. The form of the degree distribution could be affected by spatial interpolation and other pre-processing steps applied before the construction of functional networks. And the standardization of such methodological procedures will be important in elucidating the impact for anatomical resolution on the content to which power law scaling of the degree distribution is truncated at high degree. So metastable dynamics is the transitions between marginally stable network states. These transitions can occur spontaneously or as a result of weak external perturbations. And the resting state is a cognitive state in which a subject is quietly awake and alert, but does not engage in attend to specific cognitive or behavioral tasks. So the structure function relations in brain networks. How do functional brain networks emerge from structural brain connectivity? The structural maps indicate that each neural node maintains a specific pattern of structural connections with other nodes. Different nodes often have different functionalities, such as a specific response preferences to sensory stimuli. From a network perspective, the functionality of an individual neural node is partly determined by the pattern of its interconnections with other nodes in the network. Note of similar connection patterns tend to exhibit similar functionality. Although functional properties are expressed locally, they are the result of the action of the entire network as an integrated system. Structural connectivity places constraints on which functional interactions occur in the network. Structural and functional connectivity in cellular networks undergo dynamic changes. The degree to which synaptic connectivity is modified in the adult brain is highly debated. Although some evidence suggests that mammalian cellular networks are continually remodeled, other evidence suggests that some synaptic spines are stable. Changes in neuronal connectivity necessitate homeostatic mechanisms to ensure functional stability. Multi-electrode recordings data suggests that cellular functional networks exhibit transient synchronization and metastable dynamics. These changes occur within seconds, and it seems possible that even more rapid transitions and network reconfigurations may take place. These observations of relatively slow structural modifications accompanied by faster changes in functional linkages pose major unresolved questions regarding functional stability in neural circuits. It is currently unknown whether large-scale cortical networks in adult brain undergo structural modifications on fast timescales. Most of the changes that have been observed were associated with aging, disease progression, and experience-dependent plasticity. By contrast, patterns of functional connectivity between brain regions undergo spontaneous fluctuations and are highly responsive to perturbations such as those that are induced by sensory input or cognitive tasks on a time scale of hundreds of milliseconds. These rapid reconfigurations do not affect the stability of global topological characteristics. On longer time scales, the seconds to minutes correlations between spontaneous fluctuations in brain activity form functional networks that are particularly robust. For example, a set of posterior medial, anterior medial, and lateral parietal brain regions comprises the default mode network. The persistence of functional networks associated with the brain's resting state provide an opportunity to investigate how much of the pattern of functional connections is determined by underlying structural networks. Observations from the single cortical slice structural imaging fiber bundles linked components of the default network and direct comparisons of structural and functional connectivity in the same cohort of participants suggest that structural connections are highly predictive of functional connections. Structural connections are highly predictive of functional connections. Indirect interactions can account for additional functional linkages. Such indirect connections can lead to discrepancies between structural and functional connectivity. However, current evidence suggests that topological parameters are generally conserved between structural and functional networks. Computational models offer a complementary method to investigate structural functional relations in brain networks. Several studies have used empirically derived structural brain networks to specify the wiring between neural units, which then results in spatially patterned dynamic interactions. See figure 1. That's right here. We got the brain, and we've got a structural brain network and some functional brain network. The covariant structure of the endogenous activity generated by these neural units yields a functional network that can be generated by computer simulations or in some cases analytically. This strategy has been used to determine the effects of structural topology on functional networks and dynamics. Simulation studies of large-scale cortical networks demonstrated the emergence of complex spatiotemporal structure in neuronal correlations at multiple timescales, as well as realistic patterns of modeled bold resting state correlations that depend on the topology and time delays in the structural coupling matrix. The topology of structural and functional networks was identical when functional connectivity was estimated from long time samples, but functional networks estimated on shorter time samples or at higher frequencies were less strongly constrained by the structural wiring diagram. Likewise, it has been shown that modularity of the structural networks can determine the hierarchical organization of functional networks and may be important for generating diverse and persistent dynamic patterns. In a computational model of phase synchronization between coupled neurons, patterns of local versus global synchronization were found to depend strongly on the balance between high clustering and short path length in the connectivity of the network. Computational studies also began to determine the effects of functional activity on structural topology. It has been shown that initially random wiring diagram can evolve through synaptic plasticity to a functional state characterized by a small world topology. The most strongly connected nodes and by self-organized critical dynamics. These findings indicate that the brain's structural and functional networks are intimately related and share common topological features, such as modules and hubs. See figure 2. Although most studies provide support for the idea that structural networks determine some aspects of functional networks, especially at low frequencies over long time periods, it is less clear how the structural topology both supports the emergence of fast and flexible reconfigured functional networks and is itself remodeled by function-related plasticity on slower timescales. This is figure 1, the computational modeling of structural and functional brain networks. Computational models have been used to demonstrate how dynamic patterns arise as a result of interactions between anatomically connected neural units. Shown is how such a model is generated and used. A structural brain network this time structural brain network derived from anatomical data serves as a matrix of coupling coefficients that link neuronal nodes, the activities of which unfold through time. This time evolution is governed by physiologically motivated dynamic equations. The example shown, the surface of the macaque cortex was subdivided into 47 nodes, and the structural brain network linked these nodes was compiled from anatomical track tracing data. The dynamic equations were derived from a model of large neuronal populations, the parameters of which were set to physiological values. Data from computer simulations that yield functional brain networks. Such networks are derived from measures of association between the simulated time series. For example, an information theoretical measure such as mutational information computed on voltage time data or cross-correlation in neural activity that are computed from simulated blood oxygen level dependent bold data. These matrices can then be thresholded to yield binary networks from which network measures can be derived. The fact that both structural and functional networks are completely specified in the model facilitates their comparative analysis. The structural brain network panel is reproduced with permission of reference 109. So we've got anatomical track tracing of the macaque brain. There's a structural brain network, coupling matrix and neural dynamics. And then you got simulated bold time series and simulated electrophysiological time series. And there's the functional brain network, mutual information, and cross correlation right there. There's this diagram here of how these graphs are generated from the correlational matrices of the structural brain network and the functional brain network. And here we have those connection diagrams. All right, so clinical and translational aspects. Since the work of pioneers such as Wernicke and Maynard and de Jurgen, it has been appreciated that many neurological and psychiatric disorders can be described as disconnectivity syndromes. The emergence of symptoms of functional impairment in these disorders can be theoretically related to the disruption or abnormal integration of spatially distributed brain regions that would normally constitute a large-scale network subserving function. Using network properties as diagnostic markers, one application of complex network theory in this context is to provide new measures to quantify differences between patient groups and appropriate comparison groups. Several studies have reported that the parameters of brain 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 networks derived from fMRI, e.g., or structural MRI data, are altered in patients with schizophrenia or Alzheimer's disease. If an fMRI study of AD clustering was significantly reduced at a global level in whole brain networks operating at frequencies below 0.1 Hz and at local levels in both hippocampi, the global clustering was able to discriminate AD patients from age-matched comparison subjects with high specificity and sensitivity, implying that loss of small world network properties might provide clinically useful diagnostic markers. In comparable EEG study, path length in beta band 1535 Hz functional networks was significantly increased in patients with Alzheimer's disease. Importantly, the variability of cognitive function among both control and IID groups was negatively correlated with path length, providing direct evidence that functional network topology can be related to variation in cognitive performance. A third MEG study of resting state functional networks confirmed a degradation of small world attributes in patients with AD and suggested that this effect is due to disease-related changes at highly connected network hubs. However, in a fourth study that used between subject covariation and regional measures of cortical thickness to infer anatomical networks from a large structural MRI dataset, global clustering was significantly increased in patients in AD, and there were abnormalities in the topological configurations of crucial high centrality nodes in regions of the multimodal association cortex. Schizophrenia has been investigated by comparable approaches. The economical small world properties of low frequency functional networks derived from fMRI data were shown to be impaired in patients with schizophrenia. Studies that used EEG to measure synchronization likelihood or nonlinear interactions between cortical nodes found that the clustering and path length parameters of functional networks were closer to their values in random graphs for patients with schizophrenia. Anatomical networks derived from interregional covariation of gray matter density in structural MRI data showed differences in the hierarchy of associativity of multimodal and transmodal cortical subnetworks in healthy volunteers, suggesting that these major divisions of the cortex may have developed according to different growth rules or evolved to meet different selection criteria. Topological abnormalities in people with schizophrenia included a reduced hierarchy of multimodal cortex. Figure 3. The schizophrenic group network also exhibited relatively long physical distances between connection regions compatible with inefficient axonal wiring. Thus, there is a convergent evidence from methodologically disparate studies that both AD and schizophrenia are associated with abnormal topological organization of structural and functional brain networks. However, there are also inconsistencies between existing studies. For example, clustering is reportedly increased in the structural networks, but decreased in functional networks in Alzheimer's, which might be attributable to clinical heterogeneity in the patient groups, as well as the differences in imaging and analytical methods. Some of these differences may perhaps be resolved by studies combining network measurements on structural and functional neuroimaging data acquired on the same patients. It also seems likely that evidence for network abnormalities in other neuropsychiatric disorders in conditions such as epilepsy, attention deficit hyperactivity disorder, or spinal cord injury will accumulate as the disorders are increasingly investigated from this perspective. Now, assortativity is a measure of the tendency for nodes to be connected to other nodes of the same or similar degree. So rich clubs, basically. And an endophenotype is a quantifiable biological marker of genetic risk for a neuropsychiatric disorder, or anything, I guess. All right, so let's take a look at some of these figures. Let's look at figure 2. We've got a cellular functional network there, and we've got module 1, and then a hub node in module 2. The hub node connects module 1 to module 2. And then we got the whole brain structural network there. And there's that hub node again. So figure 2, we have the cellular and whole brain networks demonstrate consistent topological features. The top panel shows a cellular functional network constructed from multi-electrode array recordings made in the anesthetized cap. Each node represented by a circle corresponds approximately to one neuron, and the connections represented high functional connectivity between neurons. The different colored nodes constitute separate clusters or modules. The plots in each circle illustrate cellular responses to stimuli of different orientations, and the circle size corresponds to the degree, the number of functional connections in each node. The bottom panel shows the whole brain structural network constructed from histological data on the macaque cortex. Each node corresponds to a brain area, and the connections represent axonal projections between areas. The network has two main modules, where shown here with yellow and gray circles, corresponding to mostly dorsal and ventral visual regions, respectively. Both networks exhibit small world attributes of high clustering and short path length. Yes, this is showing the functional and structural network similarities. See box 3. So both have exponentially truncated power law degrees and associated with the existence of high degree hubs, V4 in the anatomical network, and both have the community structure characterized by sparse connectivity between modules. That is, each node is enclosed by stippled lines and linked by hubs. Which are nodes circled in red. The ATIV is the anterior infratemporal ventral area. CITD is the central infratemporal dorsal area. CITV is the central infratemporal ventral area. DB is the dorsal preludiate area. FEF is the frontal eye field. FST is the floor of the superior temporal area. LIP is the lateral intraparietal area. MT is the middle temporal area. PIP is the posterior intraparietal area. PITD is the posterior infratemporal dorsal area. If PITV is the inferior temporal ventral area, PO is the parieto-occipital area. TF area, TF, TH area, TH, V1 to V4 visual cortex, 1 to 4 VIP, ventral interparietal area, VOT, ventral occipitotemporal area, VT, ventral posterior area. The top panel was reproduced with permission from reference 86, the bottom panel, reference 49. Now, for figure 3, we have some disease-related disorganization of brain anatomical networks derived from structural MRI data. In both parts, the nodes of the circles represent cortical regions from connections represent high correlation and gray matter density between the nodes. Nodes are arranged vertically by degree or separated horizontally for clarity. Clarity of representation. The numbers indicate approximate Brodmann area, and the prime symbols denote left-sided regions. The clustering coefficient of each node, a measure of its local connectivity, is indicated by its size. Nodes with high clustering are larger. A. The brain anatomical network of a healthy volunteer has a hierarchical or network constructed from a hierarchical organization characterized by low clustering and high degree nodes. Low clustering, high degree, healthy volunteers. There, B, low clustering, healthy volunteers. Now, B, the equivalent network constructed from MRI data on people with schizophrenia shows loss of this hierarchical organization. High degree nodes are more highly clustered. This figure is reproduced with permission of Ref 124. Well, that's pretty cool representation. All right, so understanding the pathogenesis and treatment of brain disorders from a network's perspective. Many psychiatric disorders are highly heritable and are likely to be to represent the clinical outcome of aberrations in formation of large-scale networks in utero. Or during early postnatal life. Measures of network topology may be worth investigating as intermediate phenotypes or endophenotypes that indicate genetic risk for neuropsychiatric disorder. However, network metrics have not yet been adopted for this purpose. A study of healthy twin pairs has shown that classical small world metrics on brain functional networks derived from EEG data have high heritability. All right, small world. A necessary prerequisite for their candidacy as disease endotypes. Another study of graphtheoretical measures on anatomical networks derived from interregional correlations in cortical thickness MRI measurements on sample of normal twins, singletons, and singleton siblings of twins showed that genetically determined frontoparietal networks had small world properties. Network metrics are arguably more attractive as intermediate phenotypes than local measures of brain disorganization. because computational models of network development are often available to test mechanistic hypotheses for how an observed profile of anatomical or functional disconnectivity in mature network might have been generated by earlier developmental abnormalities. Another example of how empirical and computational approaches can be usefully combined is provided by studies that have lesioned anatomical or functional network models, for example, by deleting nodes or connections to explore how acute or focal damage could affect the overall performance of brain networks. Networks can be lesioned by random deletion of nodes or edges, or by targeted attack on the highest degree nodes in the network. The vulnerability of network to damage is assessed by comparing its topological or dynamical behavior after the lesioning to its intact behavior. Different network topologies confer different vulnerabilities to the effects of random or targeted attack. Um, for example, scale-free networks are robust to random error, but highly vulnerable to deletion of network hubs. Brain functional networks with an exponentially truncated power law degree distribution were found to be less vulnerable to attack than scale-free networks. In an anatomically informed computational model, deletion of hub nodes produced widespread disruptions of functional connectivity that were consistent with effects reported in focal human brain lesions. Computational lesioning of network models was also used to explore the functional consequences of a gradual and precisely specified disease process, the elimination of long-range projections, and the sprouting of short-range connections in a model of epileptogenesis in the rat dentate gyrus. The topology of the normal or non-epileptic dentate gyrus became relatively overconnected and dynamically hyperexcitable as a result of cellular changes previously described in relation to temporal lobe epilepsy. Other studies of models of temporal lobe epilepsy have shown loss of small world topology in cellular networks using hypersynchronized bursting. and have shown a variation of small world topological and synaptic properties of a computational model can cause transitions between normal bursting and seizure bursting. It's also conceivable that network analysis can be used to further our understanding of the therapeutic effects of pharmacological or psychological therapies. Dopaminergic drugs can modulate measures of functional connectivity in animal and human fMRI and MEG, magnetoencephalography recordings, suggesting the drug effects might be quantifiable in terms of altered functional network topology. This has been confirmed directly in a study which demonstrated that dopamine D2 receptors antagonists impaired the economical small world properties of the human brain fMRI networks. Future work might include efforts to using graphtheoretical measures to quantify how therapeutically effective treatments remediate topologically suboptimal network configuration in patients. Conclusion and prospects. It is clear that certain aspects of the organization of complex brain networks are highly conserved over different scales and types of measurements across different species and for functional and anatomical networks. The archetypical brain network has a short path length associated with high global efficiency and information transfer, high clustering associated with robustness to random error, a degree distribution compatible with the existence of hubs, and a modular community structure. Furthermore, anatomical networks are sparsely connected, especially between nodes in different modules, and the wiring length, the physical distance between physical distance with connection spans, the connection span is close to minimal. This profile of topological and geometric properties is typical not just of brain networks, but also many other complex networks, including transport systems and intracellular signaling pathways. Why might this be so? A parsimonious hypothesis is that many spatially embedded complex networks have evolved to optimize the same set of competitive selection criteria: high efficiency, of information transfer between nodes at low cost connection costs, or to achieve optimal balance balance between functional segregation and integration that yields high complexity dynamics. If wiring costs were exclusively prioritized, the network would be close to a regular lattice. Whereas if efficiency was the only selection criteria, the network would be random. The existence of a few long-range anatomical connections can deliver benefits in terms of efficiency and could arguably account for the evolution of economical small world properties in brain networks at all scales. This hypothesis needs to be more directly explored and tested, perhaps using evolutionary algorithms and computational models of brain network selection. A key issue for the future will be to consolidate our understanding of how functional networks interact with their structural substrates. At low frequencies or over long time periods, there are reasons to expect the functional network should be highly isomorphic with underlying structural networks. But clearly, function can be adaptive over much shorter timescales than structure. We need to understand more about the non-stationary or metastability of brain functional networks. How does functional network topology change over time? Do functional networks exist in a dynamically critical state at some or all frequency intervals? What constraints on the itineracy of network dynamics are imposed anatomically, and how does the long-term history of functional activity in a network feed back on the development and remodeling of the anatomical connections between nodes? A related question concerns how the parameters of complex brain networks relate to the cognitive and behavioral functions. One can make an intuitively reasonable claim that high clustering favors locally specialized processing, whereas short path length favors globally distributed processing. But the empirical evidence is currently almost non-existent. This will probably be a key focus for future work that might be combined with further studies of clinical disorders or cohorts at different stages of normal development. The emerging field of complex brain networks raises a number of interesting questions and provides some of the first quantitative insights into general topological principles of brain network organization. The fundamental growth in the statistical mechanics of complex networks and the power and elegance of graph-theoretical analysis suggests that this approach will play an increasingly important part in our efforts to comprehend the physics of the brain's connectome.